Hi everyone, my name is Thalita Ferreira dos Santos and I'm going to present my work titled The Structure of Ant-Plant Mutualistic Networks in terra-firme Forests in Central Amazon. My advisors are Fabrício Bácaro and Tiago Iso. Environmental and geographic gradients have been explored to understand species diversification in tropical ecosystems. The Amazon comprises a rich diversity of interactions between species related to availability of resources and habitat conditions that are crucial for their coevolution on a local scale. Among the mutualistic interactions, we have the well-known ant-plant associations. In tropical forests, the ant community represents about 50% of the arthropods diversity that have interaction with plants. When ant protection associations range from facultative and generalized, such as plants with extrafloral nectaries, to obligate and highly specialized, such as ant gardens and myrmecophytes. Studies of ant-plants mutualistic interactions have used the networks approach for two or more trophic levels. In these networks, nodes, here in green-red color, represent the species. Lines represent the presence of interactions, and the width of the lines indicate the interaction frequencies. Here we have three types of networks. In random networks, there is no community-level patterns of specialization. In a modular network, species within a model can interact exclusively with species from the same model by preference from a group. In this case, we have three distinct models. In strongly nested network, species with a lower degree are here as generalist species interact with a subset of the species with a higher degree interact, called in specialists. Considering the different interactions and metrics observed, our question is how the strength of obligate and facultative interactions affects the ecological networks in terra-firme forests in central Amazon. We have two hypotheses. The network structure for the anti-mimicophytes is more specialist. Wherefore, these networks will be more compartmentalized. Besides this, the network structure for ants EFN interaction is more generalist, wherefore these networks will be more nested. The work was conducted around the Coieiras River, which is located close to Black River Basin in Manaus. Ants and plants were collected during the dry season in 7 plots of terra-firme forests with a minimal distance of 1 kilometer from each other. Each plot has 180 meters of length and 30 meters of width. In this space, all myrmecophytes and with active domaceous and plants with active extrafloral nectaries were randomly selected and sampled. Posteriorly, we collected the ants with entomological clamp and we identified as the lowest possible taxonomic level for each trophic level. We built interaction networks organized as a quantitative metrics to estimate the size of the network and the number of interactions between species The elements of the matrix were weighted by the frequency of interactions. In this case, the frequency of interactions is represented by the number of links between the pair or group of interacting species. The measure used to understand the structure and organization of species interactions at the network level was modularity, which describes the propensity of different subsets of species to interact more frequently with each other than with the rest of the species in the network. In this case, modularity was estimated using the Quambimo Modularity Index that uses observed data to measure the degree of interaction between closely related species. The modularity level ranges from 0 to 1. Another measure used was nestedness with W-noded index, which is one of the main indexes to assess quantitative data. The level of nestedness ranges from 0 to 100, being 100 perfectly nested. Moreover, we used classic new models for bipartite networks. About the results of ant and myrmecophytes interaction, in total 69 species of plants and 41 species of ants were found. The most frequent interaction was between Alomerus octoarticulatus and Itella myrmecophila, with 56 interactions, representing 41% of the total. Nests of this species have also been found in Poruma myrmecophila. Moreover, the genus Pseudomyrmex was found only in Tachygalli paniculata. But look at for network level, antimyrmecophytes shows a low level of modularity, with the value of Q 0.42, and so we didn't find an propensity of specific models in the interactions between ants and myrmecophytes. Besides that, the nestedness was 24.7. So, in EFN barren plants results, we analyzed the two main genus found in the network. Inges polaris and Crematogassa tenuicula, the first species in the network, were the most generalized and most frequent species, with 16 links and 25 links respectively. As expected, the interaction networks between E, F, and berry plants also did divide into modules with the value of Q equal to 0.31. Beyond that, the nesting value was also very low. Despite not showing a clear modular division, ants that looking for shelter in plants are known to be extremely specific in their choice of myrmecophytes. In general, Inga attracts more species of ants, as in addition to being more abundant in our study, they had greater activity in extrafloral nectaries when compared to other plant species. In fact, knowledge of mutualistic networks structure provides a community-wide perspective for species conservation. I'd like to thank UFAM and FAPEAM for the support. Thank you.